Good day. It's Wednesday, May the 12th. I'm Martin Gagel with Radius Research. Today we're joined by Colin Bletsky, COO of Mustgrow Biologics. Today, Mustgrow announced it has achieved control of root rot disease, Aphanomyces, in greenhouse trials. This, I guess, is a follow-on to your initial news release back in January, where not in a greenhouse, but in a laboratory setting, you got 100% control. Could you just give us a very brief description of the trial itself, what kind of crops it was done with, or seeds, and then I've got some specific questions to ask.
Yeah, you bet, Martin, and thanks for having me today, and first and foremost. But so as always, the way we like to target things is starting in lab first, move to greenhouse, and then eventually move to field. This is very similar to what we did in the canola clubroot issues, as well as what we're doing in Colombia with the TR4 strain and bananas as well. And we have done that. So initially you look at control and you look for control in lab, get the right amount, make sure you can kill the disease. That at right now, there are no chemical controls, there are no disease resistant varieties. There's essentially no solutions for Western Canadian growers as well as growers around the globe. And what we look for doing is just to make sure we can kill the disease first and then move it to greenhouse. So what we did is we took it with a company called Discovery Seed Labs, who is a very prominent, I guess, pulse testing and canola testing facility here in Saskatoon. And they grew up lentil plants within the pulse, within the soil. The inoculation, so the Aphanomyces was put into the soil, and then our product was added in and the plant was seeded. And what we'd like to look for is the root health, the plant health, and how our product over a multiple different ranges of doses, so different amounts of product applied to each pot, to understand how and what level of interaction we need to take to field. And it's something to follow up now when we're going to the field with Nexus BioAg in this upcoming year. It gives us a really good range and understanding of how much of our product is needed. Are we going to be economical in those fields? And what do we need to take to field so we're not wasting our time? So it was very important data, very exciting data, because being a Western Canadian grower myself, being a pulse grower with peas on my farm, if you don't have, if you do have Aphanomyces, you have to essentially, the only way to get rid of it is to not grow pulse crops. So essentially not grow lentils or peas. And for a lot of Western Canadian growers, pulse crops are a very big and important crop for their financial status, for their bottom line, as well as for their crop rotation and soil health. So it's a very important announcement. It's a great amount of data. We still have more to do, and I'm very excited about where we go from here.
All right. And when you have, like in, let's say, regular biotech, sort of human health biotech companies, they'll publish the results in a journal. Is this strictly sort of internal data that you collect, and then it helps you design the next trial? And then when you apply for registration, I'm sure the data collected from this will be part of that registration package?
That's correct. So we do everything, being a small organization, we do everything third party to have more credibility and understanding. We want to work with the people that have the credibility in the industry. And if you look at Discovery Seeds, they do a lot of work for growers right across Western Canada in canola and pulse crops. But these data points are also used to help us with the final registration in Canada. And if you look at what we need to do, taking it from lab to now to field, or greenhouse to field, it's something that's very important to understand that dose rate. And for PMRA, so the Canadian government registrations, you have to show when a product will work and at what's the lowest effective use rate, the lowest rate it possibly can work in the field as well, so that you're putting the right amount of dose. So these trials, this data point showing the great health of the plants, showing the nice health of the roots, is something that's very important and is a perfect step and what we wanted to see moving forward into the field.
And you made a comment here that there's a dose showing a positive dose response. And does that mean up to a point that the more Terra MG applied, that the better the results were, and the less Aphanomyces is in the soil?
Yeah, so what you want to show is you want to show when your product's not going to work. So you put a dose rate that's lower to show that it doesn't work. And if you look at the picture in the news release, the low dose rates showed that at that point, a very, very, very low dose, it did not work. And it showed a very good dose response as we went higher and higher with more product. To a certain point, we were actually still being very effective. Now, Part of me would like to see what do we need to do to take it even to the next level, even higher. But we have a very good dose range to show these are the areas and rates that we need to take in the field, specifically in very problem areas. And if you look at it in Western Canadian agriculture, it was back in 2019, over 90% of the pulse fields in peas had Aphanomyces. Pulse crops are close to around 9 million acres in Western Canada, roughly, give or take on an annual basis, depending on the price points. So it's a very important and large marketplace for this. And so we are looking at it. And the big thing about the testing is we now know what that range is. We now know what we need to take the field. So when we go in and start doing our field trials, essentially we're dialed into exactly what we need to work with, and now look at the dose range that's going to have effective control for growers.
And then you also made a comment in here that the control in greenhouse soil was recorded off a range of economic application rates. So you weren't trying it on 10 gallons a meter or something that nobody could afford to dose it with. You were doing it at ranges. You were, I guess, assuming some kind of sales price where you've got appropriate margins, and then so it gives the economic value for the farmer as well as you. Is that right?
That's correct. And listen, I'm also a farmer, still involved in my family farm. And if it's not going to work on an economic side, it doesn't make sense to bring it to market and spend the money. So what we're doing is always trying to work with dose ranges that fit within a grower's economics. And if you think of the importance of pulse crops, lentils and peas, and the value they provide a farmer on a per hectare or per acre basis, it is very large. And if you look at overall benefits, being able to give the opportunity for these growers to go in and actually plant or seed a lentil or pea crop where they maybe wouldn't have been able to before is extremely advantageous and economical. So we always work in that same mindset, Martin. Sometimes we may be over the price range, and we try to look at ways to bring it back and work within the system to understand it. But in this situation, we do believe we're in the right frame and the right size within the current dose range. Now, we still have to go to field and develop it further and understand it more. And that's what we're looking forward to doing with Nexus BioAg here over the next year.
All right. And so that means very soon, because we're into planting and growing season, you're going to be applying it on some fields here in the near term, and then they'll plant. And then we're talking about pulse crops here, right? Like yes. Peas and lentils, that sort of a thing?
That is correct. So Aphanomyces is typically an issue in pulses, not all pulses, but in specifically lentils, peas, so field peas, which are now very, very important when you look at the plant-based protein and all the food that's used in that side. It's also an impact in alfalfa. We're really targeting the plant-based protein areas and the seed areas. So Aphanomyces has an issue in Canada. It's having issues in the US. It's having issues in France, UK, where some very big pulse acres are grown, and even into Australia and other parts of the world, New Zealand. So it is a major issue with no chemical control. And what we try to do is provide one level of solution for growers, knowing that they're going to need good level of good genetics, good traits, and other good activities within that system, rhizobia, different things that a farmer needs to use. So we are a key part of the entire program now, we believe, for a farmer. We just have to get through this registration process and validate in the field further. And hopefully it continues to work as we see and have seen in other crops around the world.
All right. I think we've hit all the main points here. Is there anything else you want to highlight before we wrap this up?
No, I just have to say, just on a side note, I have a lot of friends from agriculture in Western Canada, and my phone has been ringing for product. I just wanted to say to growers and people listening that as soon as we do have the the ticket from the PMRA, we'll try to get this into their field as soon as we can, because there's a lot of excitement around this. There's a definite need, and we will be testing and moving this forward, and hopefully be able to show some positive results so people can, in the near future, move this into field.
All right. Colin, thanks very much for chatting today. Have a good day, and we'll talk again soon. Cheers.
Great, Martin. Thanks for having me.